Hello everyone, my name is Jie Zheng. I'm from the School of Information Science and Technology, Shanghai Tech University in China. First, I would like to thank the organizers of ISMB, ECCB for this great opportunity to present our work. Today I'm going to present the methods of KG4SL, which is to use knowledge graph neural network for the prediction of synthetic lethality in human cancers. This is a work in collaboration with my students in China and collaborators in Singapore. First, let me introduce the biological backgrounds and computational related work. Synthetic lethality is a kind of relation between genes such that if one gene is mutated or has defect, then the cell viability will not be affected. But if both genes are mutated or inactivated, then the cell will die or its fitness will be significantly impaired. Such kind of relation between genes can be used as a gold mine of anti-cancer drug targets, because in cancer cells there have been already many mutations of the genes. And therefore, if we can find a way to knock down the SL partner genes of the cancer-specific mutated genes, then the cancer cells will be selectively killed, but the nearby normal cells will not die. And therefore, SL-based drug targets can have lower side effects as anti-cancer therapeutics. Well, there have been some experimental methods for SL screening, such as based on shRNA and CRISPR-Cas9. They have limitations such as high cost and off-target effects. Therefore, computational methods have been very useful to complement those experimental methods. There are quite a few computational methods for SL prediction. One is based on statistical inference, such as the methods of a DAISY. DAISY is based on the analysis of three types of data: the copy number alterations, shRNA, and gene expression. This kind of methods is based on the prior knowledge about synthetic lethality, and therefore we call it knowledge-driven SL prediction. The second type of methods is the supervised machine learning, which is to utilize the known SLs as labels. And some traditional methods for the supervised learning have been proposed, such as the support vector machine and random forest. Recently, the state-of-the-art methods for SL prediction is also supervised learning, but is based on the graph representation learning and the encoder-decoder paradigm. One type of such methods is based on matrix factorization. This kind of methods decompose the matrix about gene relationships and other information into a low-dimensional latent space, which can help to predict SL relations. The other methods based on graph representation learning is the graph neural network, and they have achieved state-of-the-art performance. In this paper, our methods is also based on graph neural network. The main idea of KG4SL is based on the following observation. we found that most existing methods view the SL gene pairs as independent of each other. However, there are correlations among different SL gene pairs. For example, SL pairs will share molecular mechanisms such as the DNA damage repair. And therefore, we propose to utilize the knowledge graph to capture the correlation. And our methods indeed is to integrate knowledge graph and graph neural network for synthetic lethality prediction. And the problem is formulated as a link prediction in the graph neural network. Before presenting our methods, let us look at the data. We have collected the data and knowledge about synthetic lethality into two parts. One is the SL matrix, which is a binary matrix representing whether there is SL relationship between two genes. The other part is a knowledge graph called a thin-less KG about genes and relationships. From the statistics of these datasets, we can find that the SL matrix is very sparse. And in the knowledge graph, we have quite a number of nodes, which are not just about genes, as we will see shortly, and many edges. Therefore, thin-less KG is a rich wealth about the synthetic lethality. This picture shows a part of the knowledge graph in thin-less KG. There are 11 kinds of entities, including genes, diseases, pathways, and so on, and also 27 types of relationships among these entities, such as the gene regulates gene and the compounds to treat the disease, and so on. Furthermore, we represent this knowledge graph as an attribute graph. In this graph, each node and each edge will have their properties to encode the features. If we hide the attributes, then the graph can be represented as a set of triplets in the form of the head and then the relationship and the tail. Here, the head and tails are the entities in the knowledge graph. These two tables show the statistics of the entities and relationships, and they are from different sources of public databases such as NCBI and Wikipathway and so on. Next, let's look at the methods of KG4SL. This is an overview of KG4SL, which consists of 3 steps. In the first step, we take the knowledge graph and SL matrix as 2 kinds of input. And from the knowledge graph, we're going to identify a neighborhood of each gene. We call it gene-specific weighted subgraph. This subgraph will provide the context information about the gene to be utilized later to predict the gene-gene interactions. And in the second step, we aggregate the information from the neighborhood to obtain representations of the genes. And then in the third step, we will calculate the loss of the predicted genes, gene-gene interaction scores, compared with the labels, and then use optimization to minimize this loss. In this way, we can learn the parameters and captures the relationship among the genes. In the first step, we zoom in the graph in the large graph into the subgraph, and we examine these neighborhoods with different hops. The hop means the distance between the neighbors to the center gene. Because different nodes in the large graph will have different degrees, we are going to have a fixed size for the neighborhoods in order to have a balanced information of different genes. If the neighborhood of a gene is too sparse, then we are going to use duplicate sampling to achieve the equal number of neighbors. From this subgraph, then we can learn the weights of the edges to represent the importance of the neighbors to a gene. In the second step, we use the message passing to integrate the information from the different ranges of neighborhood or from different hops. In this way, we'll learn the representation of the entities in the large graph and then integrate the information from neighborhoods of different hops into one single feature vector, or called embeddings. In the third step, based on the representations of the genes learned from the previous two steps, we calculate the scores about SL. This score represents the probability that there is SL relationship between two genes. It is based on the inner product between the representations of the two genes. Then this score will be compared with the ground truth labels of SL to calculate the loss. The loss function is a combination of the cross entropy loss from the comparison of the predicted score, SL score, and the label of SLs. And the other part is the L2 regularizer. And then the loss function is minimized by using the Adam algorithm. Based on the experiments on the real data, we compare the KG4SL with the baseline models, including matrix factorization, random walk, and the graph neural network. And we can see that KG4SL achieved superior performance in all the three performance matrix. And the second best method is the GRSMF, which belongs to matrix factorization. Furthermore, we investigate whether large graph is really important for SL prediction. To do that, we compare KG4SL to a few methods that use large graph or the SL matrix alone. So one is to use the unsupervised methods of trans-E on the large graph alone, and performance is not so good. Then we apply the graph convolutional network on the SL matrix only, and the performance is better, but still lower than the methods to integrate large graph with SL. we can find that when trans-E and GCN are combined, the performance is much better than either methods alone. This indicates that the knowledge graph can really help with the prediction of SL. Then the KG4SL is even better than the trans-E plus GCN. This shows that our methods has found a more suitable way to integrate the knowledge graph and SL matrix. In addition, we visualize the representations of the genes learned from the different methods. Here on the right side is the visualization using the t-SNE method, the methods of the t-SNE. The orange dot represents the known SL gene pairs, and the blue dot represents the otherwise the non-SL or unknown gene pairs. We can find that KG4SL can distinguish these two types of gene pairs much better than the other methods. In summary, in this work, we incorporated a knowledge graph called Singless KG into the graph neural network for SL prediction. Our experiments shows that the integration of knowledge graph can indeed help improve the performance. Our methods can also be applied to other biological networks, such as drug-drug interaction and gene co-expression. In the future, we plan to enhance our work in the following aspects. First, we plan to do interpretability analysis in order to review the molecular mechanisms behind the SLs. For example, we can extract rule paths in the knowledge graph, where the paths can represent how the two genes interact through the biological network. Secondly, we would like to integrate multi-omics data, such as gene expression data from the TCGA and DepMap. And such integration can help us capture the genetic context of SLs. Last but not least, we plan to collaborate with biologists to carry out wet lab experiments to validate the predicted SLs. I would like to thank Mr. Xin Liu from my lab for help with case studies. And in the future, we plan to do more case studies to review the biological meanings of the SL predicted. And this project is supported by startup grants of Shanghai Tech. I would like to thank the reviewers of ISMB and ECCB for their very helpful comments. And thank you very much for your attention.